my plan today. I want to complete this introduction to virology. And then what I want to do, it's actually maybe spend some time on a review. You guys have an exam coming up on Monday. This is on chapters 13 and 14. I did post an email to you guys last night. I had an announcement also. So again, that's going to be next week. All right. So let me go to this chapter first. It's a basic introduction, some really important information about viruses. And then I'm not going to talk about specific diseases yet. We will start that after the exam on Monday. All right. Okay, guys. So here's what we've done so far. We've talked about bacterial diseases. We spent 4 chapters, maybe 3 weeks on bacterial diseases. So we looked at airborne bacterial diseases. We looked at food and waterborne bacterial diseases. We looked at soil and arthropod-borne diseases. And then finally, we just completed chapter 14, which was on STDs, STIs, and then contact diseases. I want to move to the other big group of pathogens. These are viruses. And what I want to establish today, basically when you leave the lecture, you say, I think I have a good grasp. If you don't already, I think I have a good grasp of what viruses really are. So we're going to go through some basic things. I'll talk about what we call subviral particles. They're called viroids, cause disease in plants. We haven't found anything to cause disease in animals or humans yet. And then talk about diseases such like mad cow disease, CJD, Kuru, fatal familial insomnia. They're not caused by infectious agents as we know it. For example, viruses, bacteria, parasites, and fungi. In fact, they're caused by abnormal proteins. All right. So here's my objective today, is to do a very basic review of viruses and subviral particles. I have my objectives listed here, but of course, again, we're going to run through this as we lecture. All right, let me get my pen. Okay, so viruses. Let's go back in history just for a little bit. Remember Pasteur? Remember Robert Koch? Remember Robert Koch? Remember Lister? So those guys lived about 150 years ago, and their life overlapped. And basically, remember Pasteur, he proposed a germ theory of disease, and then people started describing bacteria and the types of diseases they cause. Robert Koch said, you know what I'm interested in what causes a specific disease. So he develops his Koch's postulates and so on and so forth. Bacteria were beginning to be isolated and identified around 130 years ago, all right? But think about it, there was other things that were causing disease. And I'm not talking about fungi and parasites and bacteria was known. But what I'm talking about viruses. Now think about this. So let's go back in time back. If you go back to the 1890s, all right? Basically, our trek in virology started with this plant. This plant leaf. This is a tobacco mosaic. This is a tobacco leaf. Excuse me. If you notice, there are brown spots on the leaf. It turns out this is infectious. If you take this leaf and grind it up and prepare a filtrate from it, guess what? You place that on other tobacco leaves and you get the same type of symptoms. So nobody actually knew what caused this, but it was infectious. So basically, people term this, again, I don't test you too much on history or names back here, but Ivanovsky and Bujarink, they said, you know what? There's something infectious in this leaf. We don't know what it is, but we know it's not bacteria. We know it's not fungi. We know it's not parasites. There's something new that causes infection, at least in plants. Well, if you look at animals, we have similar conditions occurring. For example, in animals, we have hoof and mouth disease. So take a look at this. These are infections caused by viruses. It wasn't known then. This is the hoof, of course, of an animal. And by the way, no bacteria or fungi or parasites could be identified to that type of disease. It turns out back in maybe about 119 years ago, Walter Reed, American, of course, there's a Walter Reed Center back in Bethesda, Maryland. So he discovered the virus that causes yellow fever. All right, that's called the yellow fever virus. So well within about 15 years, 10 to 15 years, basically here's what's being discovered. There's something new out there that wasn't described before. And that thing, of course, turned out to be viruses. Now, about 15 years later, basically people discovered viruses that infect bacteria. And they are called bacteriophage. So again, every time you hear that word phage, that refers to a bacterial virus. It doesn't infect humans, but does cause disease in bacteria. And in the past 15, 20 years, guess what? We found viruses that actually infect viruses. All right, so it's kind of strange. Life gives you all these weird things, but nonetheless, they exist. Now, one of the things that actually prevented great strides in the field of virology, we couldn't culture them. So remember, all viruses are obligate intracellular parasites. They require a living cell to grow. Well, we can't culture viruses like we culture bacteria. Yes, we can culture them, but we require cell lines. In other words, living cells that you have in the laboratory or in an animal or something to grow the virus. Well, after viruses were discovered, after bacterial viruses were discovered, going back all the way from about 1900 to about 1950, really the field just exploded. Of course, electron microscopic techniques were being invented and refined. And for most of the viruses, we were able to see them using electron microscopes. So we can't use the microscope that we have in the laboratory to view viruses, most of them. Some of them are really large, but most of them, they're beyond the limit, a bit beneath the limit, of course, of our light microscope. In other words, they're in the nanometer range, all right? So I want to just establish a few things about viruses, just some basic characteristics. They have very few shapes, actually. The first type of shape we see is called helical. And again, we just looked at the tobacco leaf. If you were to collect a sample and do electron microscopy on it, you'd see a virus that has this appearance. That's a helical virus. All right, so this is one type of shape. Of course, this is a plant virus. The rabies virus that causes rabies, of course, also has a helical shape. So this is the helical shape back here. It's also surrounded by an envelope. So this is one type of symmetry. It's called helical symmetry. The other type of symmetry is called icosahedral symmetry. So again, this is what we're looking at right here. That virus, for example, with poliovirus and herpes virus, this is what they look like. Icosahedral has 20 faces, 30 edges, and 12 vertices. These are called intersects. All right, so again, we see viruses that resemble this. Herpes virus and poliovirus are viruses are prime example. And then we have viruses that do not fit into any of these two shapes, helical or icosahedral. We call them complex viruses. All right. So an example for bacteriophage, just a complex virus, we see a spiral form, but it's not really a helical virus. We see the head of the virus. It kind of looks like this, but it's really not an icosahedral. So this is icosahedron. So this is called a complex virus. For animal viruses, smallpox virus would be a great example. That is, it's a complex virus. It doesn't have a helical or icosahedral symmetry. Now, viruses range in size from very small, like 20 nanometers. This is 20 billionth of a meter, to about 250. This is the general range. In the past 20 years, of course, we've discovered viruses in single cellular organisms that are pretty large. In fact, people were surprised that they were viruses. So here's a typical Staph aureus cell. If I look like this, these viruses were as large as a Staph aureus cell. So again, in nature, by the way, this was highly unexpected. And that was only 20 years ago when people discovered the really large viruses. They don't cause disease in humans, so we're not going to spend time with them. In any case, the size ranged from about 20 nanometers to about 250 nanometers or so, all right? Now, a few other things I'd like to mention. All viruses are obligate intracellular parasites. So we've been seeing this terminology for a while now. It means it requires a living cell to grow. Now, if you took the lab last week, you cultured the bacteria on plates, you got back to the lab this week, you probably saw tons of bacteria all over the place. They don't require a living cell to grow. They grow naturally on those agar plates. But all viruses are obligate intracellular parasites. In other words, they need a living cell. Here's your cell, virus infects, you need a living cell to grow, all right? Now, a fully infectious assembled organism is is called a virion. So the thing that causes infection, like the COVID-19, we call the infectious particle a virion. Viruses can have many different shapes, as I showed you before. They're all obligate intracellular parasites, and the infective particle is called a virion. All right. Now, other things about the virus. If you look at the nucleic acid, this is the genome. It can be either DNA or RNA. It can't be both. All right. It can be single-stranded or double-stranded DNA. So you see this picture right here. What you see in purple inside this yellow shell, that is the nucleic acid. Again, it can be DNA or RNA. It could be single-stranded or double-stranded molecules. Now, if you also notice, there's this yellow shell around this nucleic acid. That is called a capsid. So capsid protects the nucleic acid. So it's made up of protein molecules, of course, and each protein subunit, you see this little white ball back here, that is called a capsomere. So capsomer are the subunits that make up a capsid. Now, some viruses are naked. You don't see an envelope around them. Like this one here is naked. You don't have an envelope. All that means there's no envelope surrounding this particle. Other viruses are enveloped, like herpes virus, for example. It has this membrane around them. Now, these membranes are like plasma membranes, the lipids, similar to what you have in your cells. But importantly, they have these spikes. These spikes are called peplomeres. That's a technical name for it. It gives a virus specificity. In other words, here's a virus. It infects a certain type of cell because the spike actually can be recognized by receptors on that cell. So those are very important parts of the viral particle. So this is an envelope virus. One that doesn't have an envelope is called a naked virus. All right. If you're just referring to the capsid and the nucleic acid, like what you see right here, forget about the envelope itself. We're just looking at the capsid and the nucleic acid. We call that appropriately nucleic acid capsid. So these are just terminologies you have to know when we talk about viruses. Because when we talk about diseases, I might say something's envelope. It has a spike or a peplomer. So a spike is a protein molecule on that membrane. It's also called a peplomer. It gives viruses, in many cases, their specificity. In other words, what types of cells they infect.
Excuse me.
All right. Yeah, go for it.
Sorry, I have a question. So, I just want to clarify, can viruses be cultured in labs?
Yeah, they can, but not like E. coli and staph. So remember, you guys did the laboratory last week. You did a 4-quadrant streak, right? And if you didn't have your lab yet, maybe you have this later this week. But you see bacteria all over the place. We can't culture viruses like that. So the way we culture it in laboratories, we can take cells from humans or animals, right? Put it on a petri plate, and then grow these cells on the plate itself. So we're not using agar like we do in the laboratory last week. And then we can take a specimen that contains viruses and then grow them inside the cell. So let me kind of blow one up for you. Here's a virion. All right, here's a huge cell. This is a living cell now. Here's a nucleus. That virus can actually enter the cell and replicate itself. So that makes it an obligate intracellular parasite. It requires a living cell to grow. So yes, you can grow viruses in the laboratory. It's not as easy for sure as growing bacteria. Are we good?
Dr. Bideshi, I have a question too.
Yeah, go for it.
How come that virus is a naked viron if it has spikes on it?
Okay, so that's a good question. So what defines naked and envelope is the envelope itself, of course. So this is like a membrane. If you take a look at your cells, right, we have a plasma membrane. Think about it that way. This virion has that plasma membrane. This one doesn't. So the spikes are just protein molecules that give a virus specificity. In other words, what it binds to. Some spikes are on the virus itself, like adenovirus. And don't worry about the names yet. And a good example for one like this would be adenoviruses. Causes many different diseases, including colds and upper respiratory tract infection. This one like HIV or herpes virus, they have that envelope around them. All right?
Okay, thank you.
Okay, cool. Other questions? All right, so let me move on. I'm going to spend very little time on bacterial viruses. I just said earlier that we have viruses that infect every living thing and viruses themselves. Viruses are not alive. Remember, they're obligate intracellular parasites. They're just DNA and protein and lipid molecules, but they cause some really bad misery for us. Think about COVID-19. All right? I want to look in general at the life cycle of a virus. In this case, we're going to focus on a bacterial virus and the terminology for you. Every time you see this word phage, that means to eat. That refers to a virus that infects bacterial cells. Think about prophage. Think about diseases we talked about before. Scarlet fever caused by a prophage. In other words, the virus has the gene to make that erythrogenic toxin. So this goes back to Streptococcus pyogenes. Think about diphtheria. It has a phage. In other words, there's a virus that invaded that bacterial cell. So bacteriophages are viruses that infect bacterial cells, have nothing to do with your cells or animal cells. All right? Now let's look at the life cycle. Imagine I have a virus or a virion. A virion is the infective stage of the virus. It can infect E. coli. Let's just pretend this is E. coli. Here's a virion that can infect that E. coli. What happens, the very first thing when we look at the life cycle, it attaches to the cell. That is called adsorption, not absorption, but adsorption. That means to stick to, all right? The next thing is this. you guys recall, bacteria have a very rigid cell wall that's made up of peptidoglycan, right? So the virus just can't enter the cell. What it does, like a needle and syringe, it literally injects its DNA into the cytoplasm of the bacterial cell. So here's the viral DNA. Here's the bacterial. This is the bacterial chromosome called a nucleoid, as you remember. The virus injects its nucleic acid into the bacterial cell. And then that DNA is replicated. In other words, you're going to produce progeny or more virions, right? You also produce proteins that is used to make up the virus. This is called biosynthesis. So we have attachment or adsorption, we have penetration, and then we have biosynthesis. All that means is that we're replicating the DNA. We're making the proteins like the capsid proteins and other types of proteins. And then when you have all the raw ingredients, here's the next stage. That virus matures. So this is called maturation, or it's called assembly. So you see what's happening? You're using all the raw ingredients here, the viral genome, all the proteins, to now make new virions. But that's still in the cell, right? So if you're a virion or if you're a virus, you don't want to be in the cell because you want to get out of there and infect other cells. Something pretty cool happens after the virus assembles itself, literally causes breakdown. Literally lyses, which means to break down, all right, the bacterial cell. And all these virions are released, and guess what? They can infect new cells and start that cycle all over again. So this is a typical life cycle of a bacteriophage: attachment, penetration, biosynthesis, maturation, and then finally release. So if you notice, the bacterial cell dies. So this is called a lytic cycle. Lysis, again, means to break apart. So this is a lytic cycle, or lytic pathway. All right? Now, some viruses, even bacterial viruses, are pretty smart. And let me give you a reason why. Imagine, check this out, right? Imagine I have 10 cells here. Let's say this is 10 cells. And I have 10 viral particles. And let's just say for every time I infect a cell, I can get 10. All right, so every time I infect a cell, I get 10 new particles. So the maximum I can get is 10 times 10, which would be 100 virions. That's not a whole lot. That's very bad, actually. Some viruses, they can do something really special. And by the way, human viruses can do this also. For example, HIV, which we'll talk about later on, and maybe next week, all right? They can do something called lysogeny. So basically, this is a state where the virus coexists with the bacterial cell. In other words, this is not happening yet, all right? So imagine how cool this is. And I'll describe the pathway in a bit. Imagine if I have a bacterial cell, here's the chromosome, and here's the virus or the viral DNA. That bacterial cell can undergo multiple divisions. In other words, now I can have 1 million cells. And so I have 1 million cells now with a virus in there. So the virus coexists along with the bacterial genome. Remember, that's called a prophage. So that word should ring a bell to you, again, with diphtheria and scarlet fever. Remember that disease is caused by an association with a virus with the bacterial cell. In this case, the viral DNA shown right here in green actually integrates into the bacterial chromosome. So now it coexists, and that virus, the bacteria replicates like crazy. Now I have 1 million cells. Now imagine if I get 10 virus per cell. So from 1 million cell, now I can have 10 million viral particles, all right? So again, you see how pretty effective this is. Whereas compared to here, I only get 100, which is not cool at all. So these are called smart viruses. They undergo a process called lysogeny. This is a genetic state where the viral genome, like right over here, this is from the virus, it actually replicates along with the bacterial chromosome. So it's a prophage. That's called a prophage. So this here would be called a prophage. So just to lay out the cycle for you, here's a virus that undergoes lysogeny. It's called a temperate virus, but that's not significant. It infects a cell. Notice what happens. That viral DNA gets chucked into the chromosome of that bacterial cell. This is called a prophage. All right? So we kind of talked about this before with those two diseases I mentioned, scarlet fever and diphtheria. And then the virus replicates with the bacterial cell, doesn't kill it or anything. But when times get rough, in other words, the bacteria is running out of food, nutrients, or UV light hits it, what happens? The virus is really smart. It pops out and then replicates itself. All right, so now I have tons of E. coli cell or whatever bacterial cell here, millions of them. You pop out, and now you do basically you go undergo a lytic cycle. So you have a lysogenic cycle or lysogenic pathway where the virus coexists with the bacterial cell, and we have a lytic pathway where the virus replicates itself, produces tons of particles, and then kills that cell. All right. Now, let me just go back to this slide again. We're going to focus all of our attention from here on human viruses or animal viruses, right? Basically, the protocol is the same, but this is the biggest problem. This is where the major difference occurs in the penetration stage, all right? So let me get my pointer. Where are you at? Come on, you're hiding. All right. So right here, you notice this? Like a needle and syringe, the virus injects its DNA into the bacterial cell. Here's what the major difference is for animal viruses, including human viruses. In this case, you don't inject DNA. The entire particle actually enters, it penetrates into the cell, all right? So we have attachment, penetration, but the whole particle enters. I'll show you pictures in a second. Biosynthesis, assembly, and release. So everything else is the same except this stage here. All right? I just want to say a little about the surface molecules that you find on viruses. This is an envelope virus, of course. You see that membrane on there. And here's the spike. Remember, the spike is called a peplomere. That's very specific. So these spikes can bind to very specific receptors on their target cells. In other words, cells in humans in this case. So this virus just happens to be HIV. All right? I'm not going to have you memorize a whole ton of this. But HIV actually has a protein called GP120. Notice this, it would bind to a receptor called CD4 receptor on T helper cells. All right? I'll talk more about this later on. But here's some examples. HIV, influenza, HIV binds to the CD4 co-receptor on T helper cells, and a few examples. I'll mention the ones I want you to know as we go along with this lecture. So again, that envelope is really important. But the peplomers, or the spikes, these are the proteins that you find on the envelope, are extremely important because it gives a virus specificity. Hey, I'm going to infect this cell, but not that type, because that type doesn't have a receptor for the proteins I have on my envelope, right? So I'm going to point out the ones I want you to know. This is called cell tropism. In other words, don't ever think a virus, one single virus can infect all your cells. That's not true at all. There are very specific cells, or maybe a group of different cell types that a specific virus can infect. And I'm just going to point out the ones I want you to know. We're going to see them later again. HIV can infect CD4 cells. These are called T helper cells or T lymphocytes and macrophages. There's a few others like microglia and monocytes, but these are the primary cell. Rabies, right here, infects muscle cells and neurons. So we're going to look at rabies next, maybe the following Monday. The other one, it's influenza virus. Influenza virus, and think about that's a respiratory disease. So these viruses don't infect blood cells. What they infect is the cells that line the respiratory passages. So those are the major ones. Again, as we go along, I mentioned a few, but for now, keep in mind, some viruses are very specific to the cell that they infect. All right? Now, we just talked about the life cycle of a bacteriophage. I want to focus now on animal viruses. So remember, DNA viruses can have DNA, they can have RNA. In this case, we're looking at DNA virus. So here's what happens. Here's that particle, right? It's an envelope virus. Here's that spike of peplomere. Here's a receptor on the cell, on the target cell. So this interacts with that receptor. And notice this, I'm not injecting my DNA as we did for bacteriophages. What I'm doing, the whole particle actually enters the cell right here. Then what happens, the capsid, this part is broken down, and the DNA is released. That DNA gets into the cell, genes are expressed, and of course, proteins are produced, and what you have is replication of the virus. So you can increase the number of progeny. Progeny is what you produce, all right? So again, the basic thing is this right over here. We have very specific interactions that occurs between the peplomere and receptors on the cell, and then you don't inject the viral DNA. That whole particle actually enters the cell. It doesn't happen with bacteriophage, right? What about RNA virus? RNA viruses do the same basic thing, but instead of replicating in the nucleus, most RNA viruses replicate in the cytoplasm of the cell. I do want to talk about one unique feature about RNA virus or category of RNA viruses. They're called retrovirus. You probably heard the term before. These are called retroviruses. The term is right here, All right, they're very unique. The best example would be HIV. So that virus is an RNA virus. This is the messenger RNA here. All right, this is the capsid. You can see it's enveloped. And of course, we have those spikes. That's a GB120 spike. Now, these are very unusual viruses. They were discovered back in actually, we've known about it for about 100 years, but it was really proven in the late 70s. But here's what they do. The central dogma of biology states this. This is how genetic information flows in a cell. You have DNA, you have genes on DNA. You guys probably know this already. Genes are transcribed, so that's called transcription. What's responsible for this is RNA polymerase. So that enzyme comes by, transcribes that gene to produce a messenger RNA. Then that messenger RNA is translated using ribosomes to produce your proteins. That's how genetic information flows in a cell. But this virus can do something unusual. It can do a reverse process. All right, so retroviruses like HIV, here's what's unique about them. When they infect the cell, they come in with this enzyme. It's already there. That enzyme, don't forget it, is called reverse transcriptase. All right, so here's what that enzyme does. The virus gets into the cell, the capsid breaks apart, the RNAs are released, and reverse transcriptase does a reverse process. So instead of going from DNA to RNA, that RNA actually is reverse transcribed. This is called reverse transcription to DNA. All right, so that's unique about to retroviruses. Retroviruses is a type of RNA virus. They possess an enzyme called reverse transcriptase. They reverse transcribe. They take their RNAs to DNAs. All right. And again, this is what happens for a retrovirus. So we're looking at something like HIV. This is the virus that causes AIDS, right? We're going to look at that disease in detail later on. But here's what it does. Here's that HIV. It binds to, remember that GP120. This is the glycoprotein that you find on the envelope. It binds to something called CD4. And again, it's all on that table. I'm just kind of rewriting it here. On T helper cells, for example, to have specific interaction with the receptors. And then the membranes fuse, this particle enters, that capsid is degraded, and what's released is the RNA. And of course, we have that enzyme called reverse transcriptase. It reverse transcribes that RNA to double-stranded DNA. That DNA gets into the cell, and of course, it chucks itself into the chromosome. When the viral DNA in animals chuck themselves into your chromosome, that's called a provirus, not a prophage. That's for bacterial viruses. This is called a provirus, all right? And then genes are expressed. You start doing the same old thing. You start making new variants, and sometimes the cells die, all right? Okay. So, learning some basic things. I know it's a lot, but they're very general definition types of stuff. And we have to establish that to talk about diseases later on. Now, many viruses, when they infect a cell, they produce what's known as cytopathic effects. Cyto refers to cells. So here's a cell. All right, here's a nucleus. Virus enters. And for some of these infections of these cells, they do certain things to them. They can produce inclusions like this, basically things that you can see in a cell. Now, I'm not going to have you memorize a whole lot of them, but I do want you to know two of them. This one here, all right, you see that this looks like an owl's eye. An owl's eyes, right? They're viral inclusions. In other words, that's where the virus is in the cell. If all you were to show me was just this slide, and I see this, I can tell you what virus it is. That is cytomegalovirus. It's a herpes virus, but I can identify that virus just by looking at what's in the cell. So no other virus would do something like that, all right? So this is called an inclusion, and that's where the virus is. That's where it's being assembled. That's called an owl's eye. So owl's eye, you link that to cytomegalovirus. Another cytopathic effect that you see is something called a Negri body. These are these dark staining bodies in neurons. So if you've taken 153, you probably talked about the structure of a neuron. We have the axons, then we have the soma. This is the body of the neuron. This is the nucleus, all right? Here's what happens. That virus gets its way from the skeletal muscle. A dog bites an individual. Virus starts moving up all the way to the central nervous system, and then it infects the cells. But this is what you would see. That's a cytopathic effect. It's called a Negri body. Negri bodies are very specific to rabies virus, all right? So those are the two examples I'd like you to know. There are others if you're interested. Here's a nice little table for you. All right, so here's what I want you to start thinking. We'll be looking at viral diseases pretty soon. When a virus infects a cell, what can happen? So first of all, we can have a productive infection. All that means is the virus infects the cell, it replicates the cells, it lyses the cells, and then you produce more virus. All right? So that's like we saw for earlier. That's a productive infection. So think about chicken pox. When people have chicken pox, they have these lesions on the skin. The virus is replicating. All right, you see this looks like a little blister, but there's millions of viral particles in these blisters. That's a productive infection. You're producing more virions. Viruses can also do this. They can infect an individual, maybe go through a productive infection, but they can end up as being latent viruses. Latent means they're just hanging around. They're there. You haven't destroyed them. Your immune system haven't wiped them out. They're still there, and they kind of coexist with the cells, depending on where they're at. And given the right scenario, they can pop out and then undergo a productive infection. So again, with latency, the virus is still there. It's not dead per se, but it's hanging out with cells. Given the right situation, what happens? They can now pop out to cause a productive infection. Let me give you an example, maybe something that you can relate to. Think about chicken pox. Very few people have chicken pox in this country in this day and time. Typically, there's between 9 to 15,000 cases. Before 1995, 4 to 6 million cases. Why? Vaccines. Vaccines are really, really critical for many viral diseases, to prevent many viral diseases, all right? So someone can have chicken pox. Maybe you had chicken pox. I had chicken pox when I was 26. Usually you get this when you're a kid, all right? Some people think I'm still a kid, but nonetheless, I got that when I was 26 years old. So I had all the lesions on my skin. And of course, I was highly infectious. Here's what can happen. That's a productive infection, right? Because you produce tons of viral particles. But what happens in some individuals, that virus, chickenpox virus, hangs out in the neurons, in the ganglia, all right? And of course, given the right stimulus, it can pop out again to cause a productive infection. Well, that's latency. When the virus is just there, it's just hanging out, not causing signs and symptoms of the disease, pops out because of certain triggers. Then you see infections again on the skin, like in shingles. So people who have chickenpox, a good percentage of them can actually have shingles later on. Why? It's because the virus is hanging out in them. They're literally hiding. And that's called latency. All right? Okay. Now, there are viruses that can cause cancers. And let's look at the term. If people are in the oncology ward in a hospital, basically that's a cancer ward. Oncogenic viruses are viruses that can induce cancers. And again, for a long time that was debated, but nobody argues about this anymore because we know this to be true. So this was determined about 50 years ago. Viruses do cause cancer. Some of them, not all of them. So let's talk about cancer. Cancer really is an unregulated growth of cells. So I have a cell back here. Let's just say this exists like this forever. Something happens to that cell. It tells that cell to replicate like crazy. So now I have unregulated replication of that cell. That's what a cancer is. And of course, this can become a tumor, a cancerous tumor. Many different things can cause this. For example, chemicals, ultraviolet light. People also have genes, mutated genes that can lead to this condition. But of course, viruses can cause cancer also. I'm going to just mention two for now. There are a few of them later on we'll talk about. In fact, I'm going to mention three right now. One is called the Epstein-Barr virus, all right? You probably heard about infectious mononucleosis. It's also called the kissing disease. You can find this in the saliva. It's pretty stable in the saliva for a while. But again, that's a herpes virus, by the way. Burkitt's lymphoma is where the virus infects B cells. This is a type of lymphocyte. And here's an individual. See, it's primarily on the African continent. That's a tumor, all right, of the jaw. B cells are infected. Probably the most significant one that you might know of is called HPV. That's the human papillomavirus. And the biggest problem we have with HPV is in women. It can cause cervical cancer. So this is a cervix. This is a person with cervical cancer. That's induced by a virus, HPV. All right? Now here's a list. And again, the ones I want you to know is HPV. All right? We talked about Epstein-Barr virus. So these are the ones I mentioned: HPV, Epstein-Barr virus, And then the last one I want to mention is called HHV8. This is also a herpes virus. So Epstein-Barr and herpes virus 8, HHV8, that's human herpes virus 8. If ever you work with AIDS patients, so they have HIV and they have AIDS, and you see these brown patches on the skin, that is called Kaposi sarcoma. So again, this is an oncogenic virus, right? So we have HPV, EBV, Epstein-Barr virus, and then herpes virus 8, HHV8 specifically, causes Kaposi sarcoma. Others also cause cancers, but I'm not going to stress those, all right? So these are the three ones again: HPV, Epstein-Barr virus, which causes infectious mononucleosis and Burkitt's lymphoma, and then it's linked to Hodgkin's lymphoma also. We're not sure if it actually causes this. It probably doesn't. We see a good link between them, all right? And again, this is not for you. Don't worry about this. Don't worry about these. We're going to talk about these viruses later on. Don't worry about HPV yet. We're going to spend some time on HPV. And now we get to these tables, right? Don't memorize these names. Every time you see a word viridiae, that refers to a family of viruses. So many different species in this different group. It's a nice table, though, because it tells you the name of the virus or the viral family, all right? It tells you the type of nucleic acid, tell you the shape, which I want to ask you to memorize, and it tells you if it's enveloped or naked, all right? Tells you the size, and of course, it tells you the types of diseases these viruses cause. But we will talk about these diseases as we go along. Here's a nice little concept map for you. After we get done with our study of viruses, you can take a look at this. You say, oh, here, DNA viruses, these are examples, and these are the diseases they cause. Here's RNA viruses, these are examples, and those are the types of diseases they cause, all right? This is a table to show you the types of medications we use. They're called antivirals, not antibiotics. Antibiotics, as you recall, chapter 10, they're specific to bacteria. Antivirals are specific to viruses. And then, of course, antifungals are specific for fungi. And then, of course, antiparasitic drugs are specific for parasites like worms. All right, don't worry so much about these yet. Okay. Typically, what they do, I'm just mentioning a couple of things. You probably saw commercials for acyclovir or Zyrovax, valacyclovir or Valtrex. This is a common one. Ribavirin. This is Virazole. Here's what they do. They kind of inhibit replication of the virus. All right. So literally, they inhibit these viruses from replicating. Why? It's because it messes with their polymerases. And again, I'm not going to get too much into mechanisms here. When we talk about HIV, I'm going to get back to some drugs that we use for them. All right. So we're winding down with a few other things about not viruses, but what we call subviral particles. The best example are called viroids. You know, these are not true viruses. What they are, they're just RNA molecules. But we find them in plants. We haven't found any in humans yet, or animals in particular. But we know this. Check this out, all right? And again, this doesn't cause disease in humans or animals as far as we know. Here's a healthy tomato plant. You inject it with just this piece of RNA, and this is what happens, all right? So it is pathogenic. You do see some pathological effects back here, smaller growth of that plant. So it is, at some point, I think we will find viroids in humans or animals, and they might be linked to disease. It takes a while for us to discover these things, but nonetheless, viroids, as far as the ones that we know, there's just short pieces of RNA molecules, single-stranded molecules that fall back on itself like this. And then, of course, they can be pathogenic to plants. So far, again, we haven't seen viroids in animals or humans, but I'm pretty sure at some point we will. All right? Now, the last topic on the list is prion disease. And again, if I mention to you this term, if I say to you mad cow disease, I'm sure you heard that. If I say to you CJD, you probably haven't heard about that before. If I say to you kuru or scrapies or maybe wasting disease, you probably haven't heard about those diseases before, but you've heard about mad cow disease. It's all the same. All right, so the mechanisms are based on the same principle. So this was actually proposed by Stanley Prusiner back in the 80s. He's at UC San Francisco. He said, you know what? I'll tell you the story in a bit. He said maybe infectious protein molecules can cause disease like this. Now, these diseases I'm going to be talking about, prion diseases, we can classify them as, check this out, Transmissible, it can be transmitted from one individual to another or from an animal to a human, like in a mad cow disease. Transmissible spongiform encephalopathies, or TSEs. Encephalo refers to the brain. All right? So again, there's a problem that's going to occur in the brain. Take a look at this. Why is it called spongiform? Take a look at these holes in the brain. So every single area back, that's not normal. Here's what we're going to find out. Proteins precipitate. Prion proteins precipitate here. They become insoluble. They form these aggregates, and they destroy the tissues. All right? There's no cure for this. When somebody has mad cow disease, at some point they will die. All right? When somebody has Kuru, at some point they would die. So what actually causes this? What is this prion? What is this infectious thing that causes disease in humans? All right? Okay, so that's what I want to address. Are you guys still there? Say hello.
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 All right, good. When you guys are so quiet, it's either I'm doing a really good job, so you guys don't have any questions. I'm thinking, wow, this stuff really intrigues you. Well, I hope it intrigues you. This is a nasty disease. People will die. Doesn't show up right away. It's not a sudden disease like strep throat that shows up in a few days or so. This one can take years. And then there are other similar diseases that have this type of characteristic, but prion diseases are really nasty. All right? So let me tell you what causes this disease. Before I get to the cause, actually, let me show you some cases here. All right? Let me show you where this started. This started back in the 2030s, 40s or so. We had missionaries and other individuals who actually spent some time in New Guinea. And this is what they observed. All right? You see this kid here? He has a wasting disease. In other words, muscle wasting. He's probably in a coma or comatose. This occurred primarily in kids and mothers, or women, I should say, in general. The men, they were off hunting. But here's what happened when people died. This was a common practice in some areas in New Guinea. When somebody died, you took part of the body and you ate it. All right. In this case, it was the brain. With monkeys, they ate monkey brain. Monkeys can have the same type of problem. It turns out there was something infectious in the brain that they ate. And that's ultimately, later on, years later on, several decades later on, people identified what that was. It was an abnormal protein that we all have, by the way. All right? So this is in humans. It's called Kuru. This disease is called Kuru. All right? In animals like this one here, this is a deer or elk. I don't know if that's a deer or not, but that's called a wasting disease. Same condition, by the way. In cattle or cows, of course, it's called mad cow disease. All right? In sheep, it's called scrapies. All right? So we're establishing a few things here. There are other diseases that you find naturally occurring in humans. Same type of disease, right? Same agent. It's called Creutzfeldt-Jakob disease, or CJD. So again, in this case, somebody has a mutation in their gene for the prion protein, you can end up with this disease. If you eat meat, beef that's contaminated with this protein, then you end up with what we call variant CJD. So you get CJD, it's natural in a person's body. Variant comes from eating contaminated meat. All right? But here's the thing, all these diseases are caused by the same thing, an abnormal protein. So again, remember what we're looking at. Prion diseases, they're classified as transmissible spongiform encephalopathy. So here's what happens. So let's get to the pathology back here. We all have this protein that's called PRPC. That stands for prion protein. C is cellular. That's a normal form. All right? Imagine this is a neuron. Right over here, this is the membrane of the neuron. I have the normal form, PRPC. By the way, we don't even know what this protein does. We know when it folds abnormally, it causes this disease that we call prion disease, or TSCs, transmissible spongiform encephalopathies. So this is the normal form. The abnormal form is called PRPSC because it was first studied in goats. Remember, in goats, that's called scrapies. So SC stands for scrapies. So here's what happens. In the normal case, this protein is nice and soluble in the membrane. But this protein can misfold like this. So this is the abnormal form. See, it looks different two-dimensionally. The structure is very different. In this case, what happens, they form aggregates. So they start clumping. And of course, they start clumping, they get pretty large, they form these aggregates that precipitate. So basically, they precipitate a neuron in the brain itself, and they destroy neurons. So there's all these little spaces that you see back here, the spongiform area. This is what you're looking at, these precipitated pellets. You destroy neurons. Imagine if this is widespread in the brain. Usually it's kind of localized, but if it's widespread in the brain, you're going to lose a function, central nervous function in a period of time. Again, this can take years. In the more serious cases, maybe 6 months to a year or so, but up to 20, 30 years if it progresses really slowly, like fatal familial insomnia, same type of situation here, same protein. These individuals, they can't sleep. So they're certain part of the brain are destroyed, they just never go to sleep. And of course, meanwhile, they're losing brain function. They end up really, literally crazy. I hate to use that word, but they can't function, all right? Yeah, it's a terrible scenario. So this is prion disease called transmissible spongiform encephalopathies, a really, really nasty disease. No treatment for this. Ultimately, that patient will die. It is fatal, all right? So people with mad cow disease don't ever think they're going to get over it. It just goes downhill as these plaques, they're called amyloids, by the way, and again, that's a technical term, but as these plaques build up, all right? And just to put the fine points on this, here's a neuron, all right? We have the normal form on the membrane back here, that's what they are. And then by some mechanism, let's just pretend one of these, just in that same person, becomes abnormal. So you go from the purple form, let's just say red, to the purple or blue form. Check out what happens. The abnormal form converts the normal form to the abnormal form. So I have normal, abnormal. The abnormal form, it basically binds to the normal form and converts it to the abnormal form. So you see what's happening here? Basically, all the normal proteins on that neuron would be converted to the abnormal form. And here's what's going to happen. That abnormal form has a different structure. This is what it looks like, all right? What happens, it precipitates out in the brain. So we see these little pellets. And of course, these grow over time. You destroy neurons. Imagine if this is very critical areas of the brain. It's that disease can progress pretty rapidly. And of course, that person can die in a matter of years, all right? So that's prion disease. We've just completed a basic introduction to viruses and subviral diseases, or subviral particles, and prion diseases. So let me stop here for now and ask, are there any questions regarding what I spoke on? All right?
Yeah, Professor, I have a question.
Yeah, go for it.
So are prions kind of like latent disease, but they're kind of just always active?
No, they're not really latent disease. Once you have a focus of infection, it progresses. So it's not latent. In other words, latency simply means this. Like a virus is a latent virus. It's there, but it's not causing signs and symptoms of disease, right? In this case, that's not happening at all. Because as long as you have the abnormal form, you basically, it's a continuous process. It's slow, so it's chronic, all right? It doesn't, but it's not latent by any means. Latent means it's under control. This is not under control at all. You keep building those plaques over time. So it's a really nasty, nasty condition. I used to show a video. You know, I don't show that video anymore, and I should, but I just felt pretty bad for the older guy because he's totally out of it. He's shivering like crazy. Yeah, it's just a sad thing to look at. And, you know, the bad thing, there's no cure for it. There would probably never be a cure for diseases like this because you're destroying areas of the brain that you can't replenish. All right? Other questions? All right, guys. If you have questions, don't be afraid to ask. So even while I'm talking, hey, Bideshi, I have something to ask of you. All right, so yeah, go for it. No, there are 3 chapters on viruses, 15, 16, and 17. We're just cutting into 15. So I'm going to start talking about diseases specifically next week after your exam. So this includes 16 and 17. So we're going to be looking at viruses that cause respiratory diseases, including COVID, which is not in your book, by the way, because this is pretty new. We're going to look at dermatrophic viruses that infect the skin. and then what we call viscerotrophic viruses, viruses that infect the internal organs, like the brain, central nervous system, the heart, liver, for example, with hepatitis, and so on and so forth. All right? So we're just starting our lectures into viruses and viral diseases. Yes, you're right. So this is posted on Blackboard, right? So you have this lecture on there. By the way, I'll post my lecture that I recorded today on Blackboard too. And then over the next day or so, because I have another class in the morning, it's only an hour long. Your class is an hour and a half. So I have to post that lecture for these guys at tonight. But you would have access to it also. So I'm going to post these in time. So when we talk about, before we talk about any of these topics, you have the lecture ready to go. Here's the thing, I update my lectures. I don't like to use my lectures from 10 years ago. Sometimes I use some slides from 10 years ago, but there's new information now. For example, if you look at the slides from last year, there's nothing on COVID-19. I do want to update that little section there. So once I update it, I just post them on Blackboard for you. So yeah, they will be available by tomorrow night. But we're not going to talk about it until after the exam on Wednesday. All right? Excellent. All right, guys, just to remind you, we have an exam on Monday, 2 chapters only. I just sent an email to you guys last night to remind you. It's, again, same type of system. We have multiple choice and true and false. No fill in the blanks or no essays or anything like that. All right, you have an hour to complete that exam. And that's still being written, so it should be done over the next 2 days or so. But nonetheless, on Monday, it will be available. I do want to make a comment because you know what I've noticed? Most people do what they're supposed to do, right? So we talked about this before. You go to your lab section and you get there on time, and then you take the exam. That's fine. That's exactly what you're supposed to do. Then there's no problems. But occasionally I have a few students, they get into the program, they try to access it at the end of the class. And of course, it wouldn't let them in. And I have everything recorded because once you make an attempt to take the exam, it's recorded in our system. So people were claiming that they actually tried to enter it within the designated time. When I went back there, I saw the attempts were like 20 minutes after the deadline. Of course, you're not going to gain access to that. So you have a certain window. For the quizzes, it's like 10 minutes, right? So 10 minutes. Let's say your class starts at 2:30, just to reemphasize this. And I stated this in my email too, or emails, I should say. So let's say your class is at 2:30. I open the exam at 2:20, so that's 10 minutes before your class begins, and you still have access to it by 2:40. So that gives you a 20-minute window for you to access that. Before that time and after that time, you cannot access it. If you accessed it, let's just say at 2:39, you still have access to it because it's not 2:40 yet, right? But if you access it at 2:41, we have a problem. You're not going to be able to take the quiz. So if I see that on the record for Blackboard, I can't give you that quiz because it's unfair to the other students, as you can imagine. Good thing it happens maybe 1 or 2 times, but if it keeps happening, I can't accept giving you a quiz again just because. All right, so try to stick within that time. I send you reminders. And I think pretty much I really appreciate you guys have been following that. I realized that first exam was kind of strange because it wasn't clear where you should take that exam. In the combined section or a specific section. But everything's done, of course, in your specific section now. All right, so thank you so much for abiding by those principles. Just make things so much easier. And it's fair to everyone. All right. So you guys can, if you have somewhere else to go, you have to study for another exam, no problem. You can clock out. I have your attendance record. If you want to ask some questions, I'm still here.